9C. Male hormonal regulation. At the onset of puberty, a portion of the brain called the hypothalamus releases a large amount of a hormone called gonadotropin-releasing hormone, GnRH for short. This is released into the blood that travels to the neighboring structure in the brain called the anterior pituitary gland. The anterior pituitary gland in turn releases two hormones, luteinizing hormone, LH, and follicle-stimulating hormone, FSH. Both LH and FSH are secreted into the general circulatory system and act on the tissue of the testicles. FSH acts on the Sertoli cells of the testicles to initiate spermatogenesis, while LH acts on the Leydig cells triggering testosterone production. Recall that the Sertoli cells are found adjacent to the seminal vesicles in the testicles to support and nourish cells that are going through spermatogenesis. Testosterone is a hormone responsible for the maturation of the primary sexual characteristics that are required for sexual reproduction, such as testicle, penis, and sperm development, but also includes the secondary sexual characteristics such as deepening of the voice, facial and body hair, and sex drive, or libido. At puberty, a male's testosterone levels will increase 30-fold to a prepubescent level. As testosterone levels increase, its increasing concentration in the blood has a negative feedback effect on both the hypothalamus and the anterior pituitary gland. The hypothalamus responds by reducing GnRH production, which means less stimulation of the anterior pituitary gland to release LH and FSH. As LH and FSH production falls, testosterone and sperm production also falls. The Sertoli cells in the testicles produce a protein hormone called Inhibin during spermatogenesis that also has a feedback inhibition effect on both the hypothalamus and the anterior pituitary gland. Inhibin has the same effect as the high testosterone concentration in blood. It also decreases GnRH, LH, and FSH production, which in turn results in decreased testosterone and sperm production. As testosterone and inhibin in the blood falls, the double negative feedback effect of high testosterone and high inhibin on the hypothalamus and the anterior pituitary gland lifts, and more GnRH, LH, and FSH is produced. The testosterone and sperm production again increase. The constant fluctuation of this negative feedback mechanism ensures that the homeostatic values of testosterone in the blood is a stable 300 to 1,000 nanograms per deciliter, and the sperm count is roughly 20 million sperm per 1 ml of semen.